Hello everyone, myself Dr. Anirudha Dutta, Assistant Professor, Department of Biological Science, Adamas University. So today we are going to discuss about ion exchange chromatography. Okay, so if you remember our flow chart of protein purification, we have discussed that up to sample preparation and capturing step. So in sample preparation, what we did, we uh, discussed about the buffer and then different methods of cell disruption by which we can bring the intracellular protein outside in the buffer, and then next step would be our capturing step. So in that capturing step, what we did, uh, discuss still now, IMAC, the immobilized metal affinity chromatography, and ammonium sulfate precipitation method. So now we are going to move to intermediate purification step. Okay, so here one of the method that we are going to use is ion exchange chromatography. So why it called ion exchange chromatography? So the protein which is either positively or negatively charged would exchange the cation or anion from the ion exchange resin. Excuse me. So as you can see here, the ion exchange resin, here it is anion exchange resin, it's positively charged, and the complementary ion, that is chloride minus, they are attached to the resin because of Coulombic interaction. Now the protein which are net negatively charged would replace this chloride minus or exchange the anionic ligand like chloride minus and binds to the uh, matrix. So what is happening here? The protein is exchanging the anion and binding to the anion exchange resin. So same thing happens for cation exchange also. So in case of cation exchange, the matrix would be negatively charged and complementary ion would be sodium plus, if it is equilibrated with sodium chloride, and then positively charged protein would exchange those cations and binds to the matrix. So that's why the process is called ion exchange chromatography. And in ion exchange chromatography, the proteins get separated based on their charge. Okay, so what we learned still now, that in case of anion exchange chromatography, the matrix would be positively charged, whatever we see here, and there will be complementary anions, and those anions would be replaced by the anionic or net negative charge protein. For cation exchange, the matrix would be exactly opposite, means negatively charged, and the complementary ion would be sodium plus, and positively charged protein or cationic protein would exchange those sodium plus and bind to the matrix. So cation exchange matrix would be negatively charged, anion exchange matrix would be positively charged. So Going to our next slide. So what would be the pH of the buffer that we are going to use for ion exchange chromatography? So as we know that a amino acid has a amino group and a carboxylic group, and depending on the pH, the protonation state of the amino acid changes. So the pH at which the amino acid has net charge 0, that would be the neutral form of the amino acid, that would be the pi or isoelectric point of that amino acid. Same way, as protein is made up of amino acid, there would be a neutral form at a particular pH at which the protein would have a net charge 0. And if we go lower pH than the pi, then the net charge of the protein or amino acid would be positively charged, because the media is rich in H plus ion, and then amino group would accept one H plus and become NH3 positive. So in acidic media or pH that is lower than pi, the protein would have net positively charged. Whereas in a pH where the value of the pH is in a buffer where the pH is higher than or basic than the normal, the pi, there the media is rich in OH minus ion, and it will take the proton released by the carboxylic group. So then, and as well as the amine NH3 plus. So finally, the net charge of the protein or amino acid would be negative. So if we are planning for a anion exchange chromatography, then our protein should be net negatively charged. So the buffer of our choice would be with a pH that has higher value of pH than the pi of the protein. So then only the protein would be net negative charge. Same way, if you are planning for a cation exchange, the net charge of the protein should be positive, and then the pH should be lower than the pi. Okay, so that explains how we supposed to choose a particular pH for either cation exchange or anion exchange chromatography. So now we are going to discuss a effect is called Donan effects. Okay, so here, so even if we chosen a pH for running the ion exchange column, because of the lack of equilibration, or because of uh, the porosity, or the because of the uh, equilibration, lack of equilibration, what I said, the matrix, the micro environment in the ion exchange matrix might not be exactly same as the buffer that we applied. Okay, so in case of cation exchange as well as anion exchange, the pH in the micro environment of an ion exchanger matrix would not be exactly same as the buffer that we applied. So in general, the pH in the matrix is 1 unit higher than the surrounding buffer in case of anion exchange chromatography, and 1 unit lower in case of cation exchange. So, and that effect increases when the buffer has lower ionic strength. Okay. Now, for an, as we know, the protein is more stable, relatively more stable in basic pH compared to acidic pH. Okay. So for anion exchange, if the pH inside the matrix be 9 instead of 8, still it would not be that harmful for the structure of the protein. Okay. However, maybe for for a lot of protein, acidic pH is very harmful and for their three-dimensional structure. So in case of an enzyme, suppose if we applied the buffer 5.5, in which the enzyme is supposed to be stable, but because of this Donan effect, the matrix would be 4.5, which might be detrimental for the structure of the protein. So that's why while running a ion exchange chromatography, we have to choose a buffer that would have very high buffer capacity or very good buffer capacity. Okay, so a strong buffer that has good buffer capacity we supposed to choose so that we can avoid denaturing our protein. So what are the common ion exchange matrices that we use? So same like other matrix, the ion exchange matrix also have a inert bead that is made up of either cellulose or saccharose or sepacryl, that kind of inert polymer of carbohydrate. And then there would be a spacer arm, and then the final cation exchange or anion exchange ligand. Okay. So uh, the most commonly used uh, ion exchange matrix are DEAE or QAE. Okay, so diethyl aminoethylene, ethyl, DEAE, or carboxymethyl group, that is CM. For cation exchange, a lot of time we use SP sulfopropyl group. Okay, uh, for for CM we use for cation exchange, and SP and CM we use for cation exchange, and for anion exchange we use DEAE and QAE. So these are the chemical structure of the ligand. So now, depending on the charge and the chemical composition of those ligand group, the matrix can behave different way. Like DEAE, it's a weak anion exchanger. Okay, so protein which has very good amount of net negative charge, they are more efficient to bind this weak anion exchanger. If the protein has lower surface charge, then we're supposed to use a strong anion exchanger like uh, quaternary amine kind of thing. Okay, and also at what range of pH these matrix works, that also a good parameter we supposed to know before we choose a particular matrix. Okay, so similarly for cation exchanger, CM carboxymethyl is a weak cation exchanger, and it works at pH 5 to 10. And among the strong cation exchanger we have SP and S, methyl saccharose, methyl sulfonate, and sulfonyl group. Okay, SP, and they're good to work from pH 5 to 10, or some case 5 2 to 12. So as you can see, these matrix, they are really stable at a big range of pH. Okay, so you can choose any pH range in between this uh, functional range. Yeah, so there is another different kind of matrix that is called hydroxyapatite chromatography. Okay, so hydroxyapatite is a inorganic salt that is a complex of calcium and phosphate. Okay, so we can use that for separation of different biomolecule like protein as well as DNA also. Okay. So sometimes we also call it multimode interaction chromatography, because in this matrix protein can interact in two different mode. First, by electrostatic interaction with the charged surface of the matrix, like calcium, positively charged calcium, or negatively charged phosphate, as well as it can form coordinate covalent bond with the calcium. Okay, so there is, or form coordinate complex with calcium. So the mode of interaction, unlike normal cation exchange or anion exchange, the mode of interaction with hydroxyapatite is little bit different, because there is a additional type of interaction, that is coordinate complex formation with calcium. So but basically the method of elution or binding are same as ion exchange chromatography. Okay, so how we can elute the protein from ion exchange chromatography column? So we have to remember that the steps of running a ion exchange chromatography is almost same as other chromatographic method that we previously discussed. So the first step would be the equilibration of the column. The second step would be the binding or sample loading. Third step would be washing, and the last one would be the elution. So as we can see, the interaction between the protein and the matrix is Coulombic interaction or charge interaction. Okay, so definitely the ionic strength of the buffer around is highly dependent, like the interaction, the strength of the interaction between the matrix and protein would be highly dependent on the ionic strength of the buffer surrounding. Okay, so if there is high concentration of salt, the charge interaction would be less, and protein would not bind to the matrix. So for binding step, we have to keep a very low concentration of salt in the binding buffer, and for elution we can use high concentration of salt. So we can use two different elution method. So as we uh, discussed, we change in pH of the buffer, the net charge on the protein would change. So if we change the pH of the buffer, that would alter the net charge of the protein. So initially, suppose we are running a anion exchange column, okay, and the protein was highly negatively charged at pH 9. So if we reduce the pH to 7, the net negative charge would be reduced, and the protein would be no longer uh, bound to the matrix because of poor interaction and might get elute. So by changing the pH of the buffer, we can elute the protein from ion exchanger, as well as we can increase the ionic strength by adding more salt in the elution buffer. The salt would interfere with ionic interaction between the protein and the matrix, and that's how it will elute. So as you can see in this column chromatogram, so we plotted column volumes, or amount of buffer or the mobile phase running through the column, and concentration of sodium chloride in x-axis, y-axis, and this blue line, it indicates the OD 280. That means the absorbance by protein. So you see, in the first step is the equilibration. We did 5 to 10 column volume of the binding buffer. Then we injected our sample, okay, in the same binding buffer. So see, the sodium chloride concentration of equilibration and binding buffer is same. And then we started running the same buffer a little bit longer. So there would be a small peak. This is because of the protein that is not bound to the matrix. Suppose these protein, they have completely opposite charge, whatever it's required for to binding for the ion exchange column. It would get removed at the beginning. Then we'll start our elution or washing uh, or elution together by slowly increasing the concentration of the salt. So what we can do it in two different way. Either a step elution method, like increasing the salt concentration in different steps, or we can do a gradient elution method, or linearly increasing the concentration of the salt in a linear gradient. Okay, so here we are doing a step elution. So then we increase the concentration of salt and we run that second buffer for 5 column volume. As you can see, there was a peak of protein that got eluted in that particular salt concentration. Then in next step, we again increase the concentration of the salt one more time, and then another protein peak got eluted from the column. And finally, we did the highest salt concentration, and whatever leftover protein was bound to the column were all eluted in that particular high concentration salt containing buffer was. Okay, and then we have to re-equilibrate the column for next round of the reaction. So as we discussed, like we can do two different kind of elution. Either we can do a step elution, like binding of the running a low salt buffer during binding and equilibration, and then step by step we can increase the concentration of the salt, and depending on the strength of the protein, different protein would get eluted in the different salt concentration. The weak binders, they will get eluted in lower salt concentration or intermediate salt concentration, and strong binder, they will require higher concentration of salt for elution. Alternatively, what we can do, we can make a linear gradient of salt starting from lower salt concentration to a particular higher salt concentration with continuous linear gradient. Okay, so how these two would be different? So definitely for linear gradient, we'll get a sharper peak and better resolution, because you see, whenever we are running a intermediate salt concentration for longer time, suppose there is some protein which has a affinity such that it might get eluted in a salt concentration that is in between these two steps. So all those protein would come out in this particular salt concentration, and we won't be able to differentiate or separate those protein from each other. Same thing is true for the next step. So whatever protein that's supposed to get eluted between these two salt concentration, they would all come in one single peak, and we won't be able to separate those protein from each other. However, in case of a linear gradient, depending on the strength of the interaction, each and every protein might get separated, and we can collect them into different fraction and physically separate them and get a better purification. So another thing is important, what kind of column we should use, whether it's a narrow long column or high diameter short column. Okay, so both of them has their own advantages and disadvantages. So longer the column, there would be more theoretical number of theoretical plates, and there would be better purification or separation while going through the column length. As well as if the diameter of the column is small, then the buffer running through the column would be running more uniformly throughout the length of the column. Okay. However, if the column is narrow and long, then the flow rate through the column would be really low, and it will take long time to run a particular column. And another thing, whenever we are running a long column, if it takes longer time and run in lower flow rate, then there would be always a chance of the protein band to get diffused, and then we'll get a broad peak from this long narrow column. So for a long, like larger diameter short column, the problem is the buffer might not run uniformly throughout this diameter of the column. Okay, so that would reduce the resolution, whereas the column would run at a higher flow rate. Okay, and then it will take less time to run a particular column, and then the protein will stay lesser time in the matrix, and there is less possibility that it might get diffused, and band broadening we might not see here. So both of them have their merit and demerits. However, whenever we do scaling up for any ion exchange column, we'll definitely go for, we'll keep the length of the column same, only we are going to increase the diameter, so that the standardization that we have done with a smaller column stay similar, like we can use the similar parameter while running a scaled up column if we just increase the diameter, keeping the length of the column same. So thank you, and uh, I'll suggest the Robert Scoop protein purification book for further reading.